agriculture at Austin Community College, and I have the delight of being on the research team for Healthy Soils, Healthy Trees for what we have right now, which is our phase 4. So, this is a community science and education initiative that is clearly sponsored by Central Texas Mycological Society. And we are working with Ecology Action and Tree Folks here in Austin, and we are gratefully funded by the City of Austin Urban Forestry Grant. So we have two main goals with Healthy Soils, Healthy Trees. The first one is doing educational programs and outreach. So we want to be able to provide community workshops where people learn more about tree planting and using beneficial fungi to improve the health of their trees. And then we also do a community science aspect in two specific areas. One is the soil microbiome research, and we are partnering with the City of Austin, Ecology Action, and Tree Folks to learn more about how fungi can help drought-resistant native species of trees, or how we can help the native species of trees become more drought resistant. And then we also do some really cool science on Johnson Sioux compost research, which you'll hear in a minute that we have a really fun event coming up in April. So for this phase, we have decided to focus on research with red oak and pecan trees and their symbiotic fungi that help them become more drought resistant in our changing climate. What we are researching are commercial inoculums. So we have been doing some research, taking meetings, and building partnerships with companies that create commercial mycorrhizal inoculants. And we are also doing research on using the bioregional soil from host trees. So if you were in the meeting room just before we hit record, we were talking a little bit about the research that we did to take native soil from healthy trees, use that soil as a culture, and try to create more of that fungi. Then we can see how effective it is at improving the health of the soil. What we are not doing is trying to control the fungi of the soil in any way, beneficial or pathogenic. So we are measuring the success of the projects, of our experiments, through seedling health. We are examining the actual soil life and biodiversity, as well as the mycorrhizal associations that grow in these soil ecosystems. We are going to measure the soil health at planting time, as well as monitoring the growth in nursery settings. So I mentioned a moment ago, right now we are in phase 4. Phase 1 was what we were able to do is plant 52 new trees at Roy G. Guerrero Park. Very excited about that. Phase 2, we started a pilot at the Tree Folks Nursery where we planted 420 trees. Last year was phase 3, during which we did the research cultivating the bioregional mycorrhiza. So what I had mentioned, taking the healthy soil from trees. And then this year, we are going to measure the efficacy of both commercial inoculum and the bioregional mycorrhiza on 420 trees, as I mentioned, the red oak and pecan. So we're very excited about seeing all of those trees grow. So here's just a little bit of fun documentation from a planting day at the nursery. My friend and yours, Angel, planting acorns in some mycorrhizal inoculant. And then here we see some happy little oaks sprouting out of their happy little seed trays. Um, you know that CTMS is very much dependent on volunteer involvement. So, ways for you to get involved with that, which I highly recommend you do. One is start by scanning the QR code. We can all do that now while I'm talking. Please come and volunteer. We're going to need hands with both planting days and tree measurement. Of course, any generosity that y'all have as far as in-kind donations or monetary donations, every little bit goes a really long way with how we're able to use the funding. And also, if you are an individual that has subject matter expertise in this area, we would love to get involved. We had a great meeting this week with some of the great minds from Texas State, and they had a lot of really interesting things to say. So it's always great to have a meeting of the minds with mycology enthusiasts, people who enjoy fungal husbandry. I did it. I used that in a sentence. So please also, since you've already scanned the QR code for the last slide, scan the QR code for this side on April 12th at 11 a.m. We are going to have a Johnson Sioux bioreactor workshop at Circle Acres. This is going to be a lot of fun. It's going to be very hands-on. We would love to see all of you there. So please register for this at centraltexasmycology.org. The QR code will take you there. And thank you all so much for being here tonight. Thank you for being involved in Healthy Soils, Healthy Trees.
All right. Yes. So again, I hope you all had a chance to fill out that survey. That'll help us get some data points and help us understand where everyone's mind's at. And we're going to be Christian's talk tonight. It's going to cover a lot of these sort of misunderstandings of the Wood Wide Web as it's gained popularity amongst the general population. But I'm going to welcome Christian now. So he's an eminent naturalist from California, the co-author of Mushrooms of the Redwood Coast, as well as the upcoming Mushrooms of Cascadia. And fungi satisfy his curiosity with their endless forms, from the grotesque to the bizarre to the sublime, and provide a rich lens through which to learn about the bigger picture of ecology and evolution. He loves fish, plants, nudibranchs, moths, dragonflies, and is passionate about community science. So everyone give Christian a warm welcome, and thank you so much for joining us tonight.
Hi, everybody. Thanks for having me here, Angel, and thanks, Ellen, for that really informative catch up on what you guys are doing down there. And I just want to say how impressive and how cool it is that your club is involved in things like, uh, this project that you've got going with Tree Folks. It is not every mycological society in the country that has that kind of like really vibrant, hands-on, applied mycology arm. So you guys are lucky that you have that in your group. I just want to say I'm impressed, and I wish I lived closer so I could be involved. That's really important stuff. Also, I want to say a special shout out to Jay Justice, who I saw your name in the attendees tonight. Hi, Jay. Wish we were hanging out in person. All right, so I guess, Angel, is it all right if I just go ahead and get started? Can everyone see the screen? Yeah, Angel, is that thumbs up? We're good to go?
Yes.
Okay. All right, sweet. Okay, so what I'm going to be talking about tonight is something that many of you have undoubtedly heard about in one form or another. And this is the idea of the Wood Wide Web. If we were in person, I could ask you what you already know or what you've heard or what sort of impressions you have about this. But I teach about fungi in a lot of different settings, ranging from teaching the general public in sort of a freelance way through lectures or through weekend workshops or single events, single day events. But I also teach it at the university level in a little bit more of an academic setting, or at least I did until relatively recently. Taught younger folks. And I have sort of the opportunity to encounter a lot of people on their what they've heard about fungi, what makes it out there into the public consciousness. And it's been really clear over the past since I started mushrooming in 2004, 2005. Between then and now, mushrooms have become so much more popular across the United States. People are so much more attuned to the world of mycology, to the world of fungi, but especially in the, I would say, 10 years, maybe less, the public understanding about what fungi do, what roles they play, has really gotten a little bit inflated or a little bit warped, a little bit skewed by what I would call more narrativized media messaging than actual research. And it started to feel like I was going to have to say something about what is still just speculative or is more um, uh, something that we might eventually find out about fungi and how they interact in native systems, but that isn't actually proven yet by any sort of scientific standards of evidence, which is fine. We don't have to prove everything to talk about it. But I start to feel like people were taking for granted that some of these sort of messages that they were getting from media narratives were already well established in the scientific literature when in fact they weren't. So that was sort of the inspiration or the impetus for this talk tonight, is to sort of take a deep breath and sort of closely walk through what it is that we actually know about mycorrhizal fungi especially. So let's see here. I'm going to be structuring this by breaking it down into four sort of chapters: the background about mycorrhizal fungi, some basics. Then I want to talk about the research, what scientific consensus, such as there is, actually exists, what we know from primary literature, what we can guess at from primary literature. And then I'm going to talk about some of the ways that that research has been extrapolated or over narrativized in the media and public sort of facing um, sources of information, novels, literature, even TV. And then finally, some takeaways, what this all adds up to, in my opinion. So let's start with background. I think most of you all, it sounds like, being involved in the kinds of projects that Ellen was talking about, you probably all have a pretty good understanding about what fungi are versus plants. So fungi are heterotrophs. They're like you and I. They don't make their own food from sunlight and water and carbon dioxide the way plants do. They are non-photosynthetic, and that's really important for a couple of reasons. One, it shows us that they're not plants, but two, it makes it so that plants are very attractive partners for fungi because they are sources potentially of sugar, of photosynthetic products, that plants are able to do this magic trick of turning carbon dioxide and water and sunlight energy into sugar. And that is such a remarkable ability, physiological capacity that plants have, that if you're a fungus, evolutionarily speaking, if we're going to personify things a little bit, you are really driven to figure out a way to partner with plants to get access to some of that sugar that they're making. That would be a very advantageous thing to be able to do over evolutionary time if you can figure out an ecology that allows you to partner in an intimate way, source of sugar, a primary source of sugar, because fungi can't make it themselves. The only other way fungi get their needs met for food and for energy, for growth and metabolism and reproduction, is to do it the hard way by breaking down dead organic matter where that sugar that plants make has already been locked up into forms of organic matter like wood and dead leaves and dead needles that are much harder to break down. If you can get access to that sugar before it's processed into those more structural tissues of plants, you can potentially not have to spend all the energy that it takes to break it back down into a usable form. Um, and so mycorrhizal fungi are the groups of fungi that have learned how to do this. Learned is a term of slow acquisition of the ability over evolutionary time to partner with plants in such a way that they get access to the sugar before it's turned into wood, before it's turned into needles and leaves. And so the fungi don't have to break it back down to access that sugar. They get it straight from the source. Um, it flows down from the plant straight to the roots, and these fungi are able to intercept it there. And these are just some images showing common groups of mycorrhizal fungi that you're probably familiar with. Things like amanitas and russulas and boletes and milkcaps and coral mushrooms, some of them anyways, cortinarius mushrooms, chanterelles, tooth mushrooms, truffles. These are just some examples of mycorrhizal fungi. Not all groups of fungi are able to do um, um, this partnership with plants where they directly access the sugar in exchange for resources. And that's an important thing that I have noticed a lot of the general public gets wrong. They think that every mushroom is somehow doing this mycorrhizal relationship with trees. And in fact, it's actually a minority of mushrooms that are able to do this. It's only some groups. Many of them are very conspicuous. They're edible, poisonous, beautiful, big, and showy. And so we tend to spend a lot of time as mushroom hunters focused on these groups of mycorrhizal mushrooms, but so many other mushrooms are just decomposers or parasites. So it's an important first thing to understand that although most plants in the world have some form of mycorrhizal relationship, it's only a small percentage of fungal biodiversity overall that actually does this. So what is the mycorrhizal relationship? Well, first of all, let's just start by breaking down the word myco and rhizo. These are the Latin and Greek uh, roots, prefixes, and suffixes that mean fungus root. And the way that this looks is that a fungus isn't just the mushroom that you see above ground. That's only part of its life cycle that lasts for maybe a few weeks, but that's the reproductive structure attached to an underground structure called the mycelium, which is that cobwebby network of fibers you see at the bottom of the screen there. And that's the part that, although we don't see it all the time, it's present year round, more or less, in the soil, in the wood, underground, out of sight. And in the case that we're talking about, the mycorrhizal fungi, it's in the soil. It's in the organic matter horizon and probably the soil, the mineral soil horizon of the soil. Um, and it forms these little jackets or sheaths or little finger-like projections around the root tips of plants like you see on the right side of the screen. And that connection point where the fungus is wrapped around the roots of the tree, that is called the mycorrhizal root tip. So you can think of it like an organ that is made out of two different organisms, two different species. On one side it's a fungus, on the other side it's a plant. And this mutually created organ is the site of resource exchange, where the fungus, which has a huge surface area represented in that mycorrhizal or that mycelial network, it has so much contact with the soil and so many strong enzymes for gathering soil nutrients that it can really effectively gather water and nutrients and send those to the plant partner in exchange for sugar. So that's what's happening at these mycorrhizal root tips underground. You can actually see some of these mycorrhizal root tips if you're careful. If you go into a forest setting and you roll a log or you sort of pull up some rocks or you dig around in the soil, you can gently shake the root tips that you find, and oftentimes you can see that they're coated in fungal tissue. So they are visible to the naked eye with some care. And like I said, the basics of this relationship, which is a symbiotic mutualism, and just one point of contention here is that a lot of people in the general public think of symbiosis as inherently something that's good for both organisms involved, when in fact symbiosis just means persistent physical contact between two organisms that can include parasitism. So parasites are also symbiotic. That's also a form of symbiosis. When we're talking about a mycorrhizal symbiosis, we're particularly talking about a mutualistic form where both partners are benefiting from being in that relationship. Although, as we will see later, it's not so straightforward to tell where one form of symbiosis ends and the other begins. But for now, we're just going to assume that the basic mycorrhizal relationship we're talking about is mutualistic. So both the plant, the tree in most cases, and the fungus are benefiting from being in that relationship. The fungus sends water, phosphorus, nitrogen, other soil nutrients to the tree, or the woody plant or whatever plant it's partnered with, and in return it receives sugar. I will be sort of using carbon and sugar interchangeably over the course of this talk because sugar is mostly built out of carbon and hydrogen and oxygen, but um, it's not always the case that the carbon is being moved around the mycelium in the form of sugar. And as we will see later, that actually matters. And in many cases, we were taking it for granted in the scientific community that carbon was being moved between members of these mycorrhizal networks in the form of sugar, when in fact, carbon can be moved around in other forms as well, and they're not all equivalent. But we'll talk more about that later. It's important to understand that not every tree can partner with every fungus. There is some amount of specificity involved. Some trees are only able to partner with a very narrow range of fungi, and some fungi are only able to partner with a very narrow set of trees. So there is some exclusivity to these relationships. And although mycorrhizal relationships come in many different flavors, if we're focused tonight mostly on those sorts of mycorrhizal fungi that produce big showy mushrooms like chanterelles and russulas and boletes and amanitas, that kind of relationship, the ectomycorrhizal relationship, is about 130 million years old as far as we know from the fossil record, which is very incomplete. The fossil record is not a great way of understanding fungal evolution because fungi don't fossilize very well. So it's very likely that this relationship, its evolutionary history, is much older than that. But that is the earliest evidence we have for the existence of ectomycorrhizal fungi. All right, so let's move on now and talk about some examples of how we know what we know about mycorrhizal relationships. And this is a little timeline that contains some of the important benchmarks or some of the important milestones in our understanding as a scientific and public community about mycorrhizal fungi. So in 1881, Kamensky was the first researcher to describe that fungi and plant roots had some sort of relationship. He didn't know the nature of the relationship exactly at the time. He was just observing the existence of a physical connection, a non-random relationship between fungal fibers and plant roots underground. And a few years later, a different researcher, A.B. Frank, developed this new word, mycorrhiza, to describe this relationship. So I just want to point out that back here on this oldest end of this timeline is not very long ago. This is just a tiny fraction of human history that we've had to think about and understand and investigate through this relationship. So everything that we think we know about mycorrhizal fungi has happened just in the past a little over 200 years. That is not very much time as a community of people thinking together and researching together to develop our understanding about what these fungi are actually doing. So I want to just hold that front of mind as we talk about this. Is we're still in the early days of understanding this relationship and this ecology in these organisms. We should not pretend that we know, we don't have, we don't even have 500 years of thinking and investigating about this system. There are other kinds of biological phenomena that we have much deeper understandings of because we have been researching them for more than 1,000 years. There are early naturalists that go back to the ancient Greeks that describe certain kinds of phenomena, but mycorrhizae are not one of them. We are very new to this natural history phenomenon as a species, as a human group of people investigating this. I'm going to flash forward to 1960, so big jump there, but a researcher named Bjorkman was the first to demonstrate that carbon, once again, that's shorthand for sugar, is actually transferred in some cases from trees to a group of plants called the mycoheterotrophs. And I'm going to show you in just a moment what mycoheterotrophic plants are, but it's really important that we have this early milestone showing that yes, sugar does move from one plant species to another plant species linked by a fungal partner. So this is a really important first rung in the ladder of our knowledge about mycorrhizal fungi, because without this, we don't know for sure that this movement of sugar between two different plants is possible. So this is an important development in our understanding. In 1975, there was a researcher named Jack Harley, who I will actually go a little bit out of order in this talk, and I will address him a little bit later. But he wasn't as much of an experimentalist as he was a theorist. And what I want to highlight about Jack Harley is that he had some very insightful, complicated views about mycorrhizae that we're now coming back towards understanding that they need to be experimentally tested, and he was able to guess at the future of mycorrhizal research early on, and I think he deserves some revisitation. In 1997, Suzanne Simard, this is another really important milestone in this timeline, found that trees were able to transfer sugar, or sugar was being moved around between two different trees connected by fungi. Um, the important thing about this, as compared to Bjorkman's study, is that these are two different trees, not just microheterotrophs on one end. So a microheterotroph is something like an orchid or a blueberry plant that doesn't actually photosynthesize, whereas Suzanne Simard's work was able to show that sugar was being moved between tree partners that could both photosynthesize. And this actually led to a big wave of new interest, because it challenged the dominant ecological thinking at the time, which suggested that these trees should be in competition. There shouldn't be movement of sugar between them, because they should be competing with one another, and there would not seemingly be any evolutionary advantage to sharing resources between two different tree partner or tree species. Um, in 2009, uh, Tom Brown's and Taylor's lab showed that orchids were parasitic on fungi, some of these microheterotrophs. In 2008, Paul Stamets gave a TED talk called Six Ways Fungi Can Save the World, which was a very influential, very popular TED talk, because it was so ambitious and so seemingly utopian in some of its promises or its claims about what fungi could do for a variety of different human problems. And it influenced, I think, a lot of the way that people thought about mycorrhizal networks in the years following. Some of this sort of myco-solutionism, or using fungi to address very intractable human challenges. In 2019, so between Paul Stamets' TED Talk and sort of the modern era, the 2020s, there was a whole new wave of what I call the new wave of academic research that added a lot of complexity to what we thought we knew about mycorrhizal relationships. During this time, a lot of investigators sort of went back and sort of asked, what do we really know about these relationships, and maybe is it more complicated than the original story would suggest? And so I'll highlight a few of those papers. In 2019, Fantastic Fungi, that Louis Schwartzberg documentary film, came out that profiled Paul Stamets again, and that also made a huge splash in the public awareness. So many people tell me, who come to my classes, that they first heard about wood wide webs. They first heard about how interesting fungi are because of Louis Schwartzberg's documentary. And for that, I have to give it a lot of credit, because it turned on a huge number of people to the world of mushrooms. But I will say that I think it also left people with a lot of misimpressions, or a lot of misunderstandings about what we really know about how fungi work, and maybe got the cart in front of the horse about making some grandiose claims about how fungi work. In 2021, Suzanne Simard, that researcher who I mentioned at the 1997 timeline point, published a memoir that blended some of her research with her own personal story, life story, called Finding the Mother Tree, and it became a hit. It was a total, I think it was on the New York Times bestseller list, and it was actually featured in the TV series Ted Lasso. One of the characters was shown reading the book. And then Toby Kears's organization, the Society for the Protection of Underground Networks, also known as SPUN, was also founded in this year. And it has had a major sort of, it's been highlighted in a lot of public facing news articles and interviews. Toby Kears has been a major face of sort of the new wave of mycorrhizal researchers all over social media, really Toby Kears is one of these sort of like modern researchers in the sense that they all have a really big social media presence. There's a lot of like narrativized storytelling that goes into gaining funding, gaining public awareness, and packaging the results of their research. And then in 2023, a really important thing happened, which is that Justine Karst and some of her collaborators published the first real pushback article saying, hey, we've lost the plot. We've gotten ahead of ourselves with our public messaging about what we understand about common mycorrhizal networks and this Wood Wide Web idea. And we need to slow down and reassess and make sure that we are not getting constrained by our own desires for how the system looks, how it works, and what it teaches us about ecology at large. And this has catalyzed a lot of other people to also voice their concerns about not not getting too committed to one model of what the Wood Wide Web actually looks like, how fungi connect plants and exchange resources in forest settings. So unfortunately now, 2026, a few years later, we're still in the thick of it. This is not really, the situation is not clarified exactly. We're still in the sort of mess and tangle of trying to find a new balance, a new equilibrium of where to go from here. So I want to walk you through some of these milestones a little more closely by profiling some of the research. Okay, so common mycorrhizal networks are the sort of technical term that you'll find in academic papers in primary literature. It means the same thing as the Wood Wide Web. The Wood Wide Web was sort of a um, catchy way of talking about this that was published in the same article of Nature that Suzanne Simard's 1997 research was published. But what this means is that two or more individual plants are connected to the same fungus. Now, there may be more plants than just two, and there may be more fungi than just one, but the point is that these are networks formed by nodes of plants and fungi connected to each other in various ways. And the plants and the fungi may be of the same or different species. So you could have a pine connected to a pine, or a pine connected to an oak, or a pine connected to a orchid, a parasitic orchid. Or you could have a pine connected to an oak, connected to an orchid all at the same time. And there may be multiples of all of those partners. So these networks can get pretty complicated. The graphs of their connections can become very hard to visualize. Um, why did the recording stop? I just got a notification the recording stopped. I'm assuming that was a mistake, but just in case you want to know,
Yeah,
Angel,
that's
the
a
recording
mistake.
stopped.
Ignore that.
Okay.
Yeah.
So mycoheterotrophs, like I said, were one of the earliest examples of this phenomenon in action. So mycoheterotrophs are plants that don't have their own chlorophyll, they don't make their own sugar, they are parasitic on a fungus. That's the myco part of their name. They're heterotrophic plants, which is a really weird idea. Most plants are what we call autotrophic. They make their own sugar using chlorophyll and photosynthesis. But on the left are four examples of blueberry family plants, and on the right are two examples of orchid family plants that are mycoheterotrophs. And they provide some of the most rock solid evidence of resource exchange. Well, not exchange exactly, but one-way flow of sugar from a tree through a fungus to a mycorrhizal fungus, and then this plant comes in and it steals the sugar from the fungus. So it's actually sapping resources away from that fungus, but the fungus is ultimately getting its carbon, its sugar, from a tree that it's partnered with in a mycorrhizal fashion, an ectomycorrhizal relationship. So these plants provide us proof, strong evidence that it is possible for resources, in this case sugar, to move from one plant to another plant via a fungal conduit. There's a fungus in between. So this is what that graph looks like in a sort of simplified way. On the left, you have the orchid. On the right, you have a pine tree. And
What?
underground, you have a fungus and its mycorrhiza, its mycelium, connected to the roots of both plants. In the
Oh.
one case, the truffle in this case, the rhizopogon, is someone's got their microphone on.
Sure.
Everybody mute your mics.
I can't
I
tell
like
whose
it.
mic that is. I don't know if there's a mute all. Anyways, So in this case, this fungus is connecting both of these partners, and the tree is getting a benefit from being partnered with the fungus. So that little arc there connecting those two shows a plus and a plus sign. And in all the other cases, the orchid is getting a benefit from sucking sugar out of the mycelium underground, but the fungus is not getting a benefit. It's actually losing resources to the orchid, so it's showing a little minus sign there. This is a parasitic relationship in favor of the orchid. Now, I should say that's as far as we know right now. Maybe there's some way in which case, or in which this fungus is getting a benefit from being in relationship with this orchid. But right now, all of the evidence that we have suggests that there is no benefit. In fact, there is only a downside for the fungus in terms of losing resources to its this orchid. Um, but that could change with future research. Someone could prove somehow that perhaps this orchid is increasing the immune system of this fungus to prevent infection by some bacterium or something. I'm making up an example, but as far as we know right now, the orchid is a parasite on the fungus. And so by proxy, sort of 1 degree removed, that orchid is taking sugar from the tree, which is the ultimate producer of the sugar that the fungus received. So that's just a simplified diagram of one three-way relationship between two plants and one fungus. I'm trying to advance here and it's not working. Why is that? There we go. Okay, so in 1997, Suzanne Simard showed additionally the movement of carbon between two photoplants, not just a mycoheterotroph and a tree, but actually two different tree species, Douglas fir and birch trees, both of which are photosynthetic, both of which are mycorrhizal, and thus which can be connected by a common mycorrhizal network. One fungus can partner with both trees. And what she showed is that by radioactively labeling some carbon dioxide and tying a little bag of this radioactively labeled gas around the leaves of one tree, that the leaves could open their stomata, they could suck in, breathe in this carbon dioxide that had a radioactively labeled tracer, and then she could follow the movement of this radioactively labeled carbon from the tree attached it to down into the ground, and it would show up in the leaves of another tree nearby, a different species. So she was able to show the move carbon between two different photosynthetic plants, which was a big deal. But it should be remembered, it should be noted that in this publication, the amount of carbon that she saw moving between the trees was a very small amount. Not the kind of amount of sugar that would allow the plant to really make a living by itself using that carbon or even do very much with it, but perhaps enough to support the continued persistence or survival of that tree under extremely harsh conditions or during certain times of year where it was really struggling. But it was not a large volume of carbon that she was able to detect in this study. And also, it should be noted that the carbon was moving in sort of predictable ways, away from plants that had lots of exposure to sunlight and could make lots of sugar, and towards that were shaded and perhaps just could make less sugar. So it was following sort of simple, what we call source sink dynamics. When there's a lot of one resource in one place and little of it in another, you expect it to flow in the direction of lower concentration just by the background dynamics of physics. Things tend to flow away from areas of high concentration and towards areas of low concentration. So not as surprising there as you would expect. Or perhaps it is. More research is needed to really clarify what was going on here. But the idea that this Suzanne Simard to come up with is the idea of the mother tree. And this was sort of the central metaphor of her later memoir. The idea here being that a big established tree drops its acorns or its seeds around the base of its trunk. They germinate and become little tiny saplings that have a close genetic relationship to their mother tree that's right above them. And the idea is that by having a pre-existing network of mycelium already there in the soil, these brand new seedlings or saplings can plug into that mycelium that is connected to their mother tree and perhaps that mother tree can send them resources to give them a head start as they are growing in their first year or two of life, where they might otherwise have a hard time providing for themselves by photosynthesizing or gathering water. And so the idea here is that mother trees can preferentially advantage closely related seedlings. And potentially, this could be a way that forests maintain some sort of cohesiveness or increased forest health. But this gets complicated really quickly. One of the ways that it gets complicated is that the opposite can also happen. And we know this from the Jansen-Connell hypothesis, which has been investigated many times and comes from tropical forest settings originally, but has been found in other settings as well, where there's actually negative density dependence, where seedlings that are close to their parent tree actually die at higher rates because the parent tree doesn't just have beneficial fungi associated with it, it also has pathogenic fungi associated with it. And you can get sick from the microbial pathogens that your parent tree carries. And what the Jansen-Connell hypothesis predicts is that there's actually a sweet spot where you are a little distance away from your parent tree, but not too far away. And another thing to remember here is that there's also some just basic structural limitations that prevent seedlings from doing well near their parent tree in some cases. So, for example, a Douglas fir is what we call a shade-tolerant tree. It can grow just fine in relatively shady forest. But a pine tree is not the same as a Douglas fir. And so for some trees like pines, growing in the shade of your parent can actually cause you to die because you don't get enough sunlight to do photosynthesis and make enough sugar to grow. So it really matters which tree you're talking about. And this is another common theme that I hear people get wrong in their understanding of what the mycelium is, what common mycorrhizal networks are, how wood wide webs work, is that they basically fail to account for the fact that not all fungi are the same. They don't all behave the same. Not all trees are the same. They have very different biology, very different life history, depending on which tree you're talking about, or which fungus you're talking about, or even which setting, where in the world these trees exist, or what time of year it is. All of these dynamics are very sensitive to the exact details and the context in which you're talking about them. And it's not safe to generalize from any one research example to assume that all trees behave the same way. So yes, in some cases, there may be a benefit towards germinating near your parent tree because you will get a beneficial mycorrhizal fungus to grow with. But in other cases, there may be the exact opposite effect. There may be a negative correlation with growing near your parent tree and survival. I want to highlight this study, or the study that this graphic comes from, that was published in 2010, Song and collaborators, because it provides some of the clearest example of what the wood wide web or the common mycorrhizal network phenomenon promises. And that's communication, and I use that word very lightly here. What I really mean is signal being passed between two different plants via a fungal conduit. So what this means is that some sort of information is passed between two different plants whose only connection to one another is via a fungal connection, via a link that is in the form of a fungal partner that attaches both of their roots one to the other. And the way these authors did this study was very clever. So I want you to sort of focus on this drawing as I talk you through it. But basically what the authors did is they grew tomato plants in different combinations of connection. Let's start by focusing on that plant right in the middle of the circle. That's called the donor plant. And the donor plant is connected to the partner ring in different ways. So up on the left there's no contact with that plant at all. That's what we call the control. So that plant has no connection to the donor plant. It is covered in a bell jar, and its roots are prevented in contact with the roots of the donor. On the right, on the upper circle, there is a plant that fungal connection to the donor plant, but then the container that plant was growing in was rotated to sever those connections, so it no longer has connections to the donor plant. This is another form of a control to make sure that it's not just the presence of the fungus that is doing the ultimate effect on the plant. At the bottom right, you have a form of connection to the donor plant where only the fungus is allowed to grow through a very fine mesh, and it is the only thing connecting the donor plant to the receiver plant. And on the lower left, you have full contact between the donor plant and the receiver plant, where both the plant roots themselves as well as the fungal mycelium is allowed to grow in connection between those two plants. And so this provides 4 different forms of connection between the donor plant and the receiver plant, allowing us to determine precisely what form of connection is required for the ultimate effect to be observed. Because what the researchers did next is they exposed that donor plant to a stressor, in this case, aphids. So aphids are a little tiny invertebrate that is a pest, causes a sort of a harm to that donor plant by sucking the plant sap out of its leaves or tissues, and causes that plant to have a physiological response, production of stress hormones, closing of stomata, maybe a little wilting. And you wouldn't expect any of the receiver plants to show the same response because they were not exposed to aphids. They were also covered in bell jars. They were kept isolated. They were not exposed to the same stressor. But what the researchers found is that plants that had mycelial connections to the donor plant were able to show the same physiological stress response as if they themselves had been exposed to aphids. And what this shows is that there's some amount of information that is being detected by the receiver plant in both combinations that allowed both the roots and the fungus to be in touch, but also in the combination where only the fungus was connecting the plants together. So this is sort of proof positive that there is some way for fungi to allow plants to communicate information, whether intentionally or unintentionally, between themselves about important stimuli from the environment, in this case, a stressor. And this opens up all sorts of questions about ecology. Does this happen in forests? Remember that this experiment is happening in a lab setting, very carefully controlled, very artificial, nothing like what a real forest looks like. And that's an important thing to keep in mind. You cannot necessarily generalize results that happen in labs to what actually happens out in the wild. But this is at least something to another rung in the ladder. This shows that the proof of concept, there can be informational flow between 2 different plants via a fungal conduit. But I want you to remember that when I say that, you probably make a bunch of assumptions. I make a lot of assumptions. I made assumptions when I first heard about this research paper about how it was happening, but all the authors showed is that it happened. They did not show how it happened. They did not show what the form of information exchange was in, how it flowed through or around the fungus. All they showed was that there was some amount of information detection by the receiver plant, and the only connecting variable that mattered, or the one that mattered most, was the fungal conduit. And this is just 2 years later, Bartow and showed, or they developed that idea a little bit more, which is that yes, information might be detected by the receiver plant, but we don't know what form that was in. Some sort of chemistry, presumably, some sort of chemical, some sort of molecule was produced by the donor plant that was detected by the receiver plant, but some of these chemicals can't move through a fungus because they're too big, or they have the wrong kinds of polarity, or they're not soluble in water. And what they showed is in this diagram that there's many different routes. Maybe the chemicals are flowing in a fine film of water on the outside of the fungus, not even through the fungal tubing, not even through the mycelium, just around it. Maybe the mycelium is just providing a path through the soil that chemistry can sort of flow in one direction to another. In other words, the outer structure of the fungal mycelium can act as a wick that allows that those chemicals to flow in one direction or another, but maybe the fungus has really nothing to do with sending it. Maybe it's not actually a pipe, maybe it's more just a wick. So we really have a lot of work to do as a scientific community to prove or to show how any of these, informational chemicals are working. And it's also true that plants are very complicated in their ability to sort of discriminate between the fungal partners that they're associating with. In 2019, this is sort of one of those new wave articles that added a lot of complexity to our understanding of the mycorrhizal relationship. Laura Bogar, Kabir P.A., and some of their collaborators were able to show in a very clever experiment by growing larch saplings in the lab. Once again, this is a lab study, not a observational study out in the wild. They grew larch seedlings in different combinations of, or three different fungal partners, and they were able to determine that the larch was able to sort of have a hierarchy of preference of which fungus it would send sugar to. And the idea here is basically that not all of these mycorrhizal partners are equally useful to the host plant, that there's some sort of specialization even within this already very specialized system, where some species of slippery jacks, like the three you see on screen here, are better for that host larch. And they did in fact find evidence to support this hypothesis that that some of these species of slippery jacks are better at being mycorrhizal partners than others. And they were able to find sort of a fairly stable hierarchy of preference. So that complicates our idea of how these mycorrhizal networks might look. Likewise, they were able to find that some of these mycorrhizal conifers were so sensitive to their mycorrhizal partners that even controlling for a single species of slippery jack, what they did was they split the root systems of one little sapling and put one side of the root system into one petri dish and the other side of the same plant's root system into a different petri dish. They grew the same species of fungus on both petri dishes, but in one petri dish, the fungus was enriched with nitrogen as a sort of currency to exchange for sugar, and the other side, it was depleted. So that fungus had no sort of bartering chips, no sort of trading goods to exchange for the sugar that the plant was going to exchange. And what they were able to detect is that, amazingly, the plant was able to send carbon preferentially to the side of its root system that was in touch with the more enriched fungus. So even within its own body, the plant is able to detect which side of its roots are in touch with the more efficient or more enriched source of the better fungal partner, even controlling for a single species of slippery jack. So really, in some cases at least, really amazingly finely tuned ability to discriminate between fungal partners. So it's not true every tree is passive in this relationship. It's not like they're just sending sugar willy-nilly down into their roots for all fungi to eat freely from the buffet. There is a lot of, at least in some species, remember, not all plants are the same. At least in some species, there's a lot of fine-tuned control here. But on the other end of the spectrum, it appears that some plants really are pretty loose about how they control the flow of sugar into their root systems. And in fact, it's not the case that all plants have the same discriminatory abilities. Some of them are likely pretty insensitive to exactly where they are getting their nitrogen or where they're getting their water or soil resources, and they may be just kind of a free-for-all for the fungi that are partnered with them. And in fact, some fungal partners have been suggested that the way they make their living as ectomycorrhizal partners is, even though they're not necessarily very good at gathering nitrogen or water, maybe they're not very efficient at it, they can nevertheless make a living on a mycorrhizal plant host by just sort of hiding in a crowd. They can sort of fade into the background of other fungi that are doing that job much more efficiently, but there's still enough real estate available on the root system of a tree host that some fungi are able to sort of eke out a living by colonizing one or two root tips here and there. And I really like this idea because it resonates with me about my general sense as a naturalist about how a lot of nature works is that there is so much resources to go around in some systems that there's enough space left over for a large diversity of things to make a living, despite not being particularly good competitors with the more dominant sort of species in that system. And now I'm going to be revisiting some of the ideas that Jack Harley came up with in 1975. And I think one of the most interesting theoretical contributions that he made is we tend to think of the idea of a plant's root systems being extended by having this point of contact with that big mycorrhizal mycelial network that's in the soil. And we think of them as one sort of continuous organ. But what he said is that no, actually, maybe it's the exact opposite. Maybe the fact that the fungus forms a sheath around the tip of the root is actually more like a gatekeeper, where the fungus is preventing that root tip from accessing soil nutrients on its own terms, and it's actually sort of guarding soil nutrients from the root tip unless the root provides sugar in exchange, and it's sort of blocking that nitrogen and phosphorus resource from the root. And it's so funny to think that all it takes is a small flip in perspective. It's almost that like that optical illusion where it's like you either see the face of a woman in profile or you see, you know, two wine goblets next to each other. As soon as someone suggests to you another way of seeing the exact same data, you suddenly stop seeing the mycorrhizal sheath around a root tip of a tree as an extension of the root, as a pure, you know, happy-go-lucky mutualistic, and you actually start to see it as maybe a little bit more cutthroat, something more like a gatekeeper instead of an extension. And he actually did find some experimental evidence to support the idea that it was more of a gatekeeper, and the evidence was in the form of a delay in the exchange of phosphorus for sugar. So this was not just a constant flow of sugar from the plant to the fungus and a constant flow of phosphorus from the fungus to the plant. What he actually found was that the phosphorus was released in delayed bursts to the tree, almost as if there was sort of a gathering and then a bartering, sort of a time release in exchange for sugar. So only after a certain amount of sugar had been received would the phosphorus be sent to the tree. Now, that's not the only way of interpreting that data, but he interpreted that at least as support for the idea angle sheath was more of a gatekeeper than an extension. And he also brought up the point that sugar may not be or carbon may not be moving from one plant to another in the form of sugar. In fact, carbon can be taken in by by the fungus in the form of a sugar, changed into an amino acid, arrives at the end. It can be amino acid, a signaling molecule like a glutamate, and that is actually a protein and not a sugar that the plant could then use for metabolism or growth in the same way. So it's unless you are taking very, very careful accounting of each molecule in this system, it's not safe to assume that a carbon over here and a carbon over here are both sugars. They may change form on the way. And this is actually a really interesting study that came out just a few years ago. I was lucky enough that this came out of UC Santa Cruz, where I went to school, and I know both of these people who were involved in this researcher as mentors or people who I was able to learn from while I was there. They were working on the Joshua tree ecosystem of the Southern California deserts. Now, this is not a place you're ever going to go to look for mushrooms. These are not mushroom-forming fungi, but the mycorrhizal fungi that associate with these Joshua trees provided them with a system where they could test some of these ideas about how the nature of a symbiosis changes over the course of the life of a single plant. And what they did is they took Joshua tree seedlings and they took samples of fungi, much like what Ellen is doing, just raw samples of mycorrhizal fungi from different elevations within this plant's range, and they cross-planted them with fungi that were from upslope, downslope. And what they found is that as the trees grow over time, the fungi that they're partnered with are the exact same fungi from the beginning to the end of their study, But at the beginning, they act more like parasites. And by the end of their study, they were acting more like mutualists, providing actually benefits to the tree instead of just drawing sugar from the tree. So the same fungi can act as parasites early in the life cycle of a tree and become more like mutualists as the tree matures and has more resources that it's able to allocate towards fungal partners. The same thing happens when you take fungi from one setting, higher elevations, and inoculate it on trees from low elevations. Even though it's the same tree, they're adapted to different sets of fungi in different places. And so the context really matters. You can't just assume that because a plant has a fungal partner, that it will act as a mutualistic partner in all contexts and at all ages and at all times of year. Seasonality, age of the tree, and particular suite of fungi that's associated with really matter towards interpreting whether the symbiosis is more parasitic or more mutualistic. And it's important to remember that these are really a continuum, that there's not an obvious line where one stops and the other begins. In 2025, Toby Keir, more about in a little bit, her collaborators published a paper that I thought was really important for understanding why it's not straightforward at all. Even when you go back to that tomato experiment where there's a donor plant and a receiver plant, it's not straightforward to assume that that was some form of intentional communication. And the way they frame this is as the difference between signals versus cues. And this really matters in terms of intentionality between two different individuals or two different organisms. So for example, a signal is something like what's happening with this bower bird or this bird of paradise over on the left, where it's showing off its bright colors and it's doing a dance and it's trying to attract another bird to mate with, a partner. That's a signal. That is an intentional exchange of information from one organism to another member of its species to try and intentionally communicate its suitability as a mating partner. But you have to contrast that with something like a sneeze. Now, it might seem weird to you to even compare these two phenomena, but a sneeze actually contains information. When you're in a crowded room, and you hear someone in the back who's just constantly sneezing and coughing and sniffling, that communicates information to you as a member of the same species that you might need to move yourself away from a potential disease source. You might guard your health and your fitness, your evolutionary advantage, by being able to interpret that sneeze for what it is, which is a potential vector of a pathogen. And this is what they call a cue. Although it is information, and although it seems like someone is, maybe if an alien landed on Earth and they saw someone sneezing, they could interpret it as an intentional communication of their disease state to their fellow members, as if it was somehow a mutualistic or a altruistic way of signaling, hey, I'm sick. In actuality, a sneeze is actually just something you can't contain or suppress or control. And in fact, it might come at a evolutionary cost or an advantage to your fitness, because if you're sick enough, members of your tribe might abandon you, or they might put so much distance between themselves and you that you are left to your own defenses. So the difference between a signal versus a cue, not easy to determine when you're looking at something as different as a fungus. Their bodies are not, they're not like vertebrates. Even this little bird of paradise that we see on the screen here is so much more similar to a human. It's so much easier for us to project our understanding onto what's happening with their behavior, that we can kind of understand their biology. It is not at all easy to understand when a fungus and a plant are exchanging information. It may be more like a cue than a signal. So now I want to talk a little bit about the narratives that surround some of the common mycorrhizal network research. And I'll start with, I think, 1997, Suzanne Simard, or actually first I'll talk about Paul Stamets in 2008. His talk, 6 Ways Mushrooms Can Save the World, is really well summarized by the first line, in which he says, I love a challenge and saving the earth is probably a good one. And that just goes to show you how ambitious and how grandiose this talk was. It really positioned fungi as potentially addressing all of humans' greatest challenges on planet Earth. And it stressed the utility of knowledge, as if all we had to do was to understand how fungi work, and then we could use them to solve our problems. And I think that really colored how a lot of people in the subsequent years understood mycorrhizal research. They framed it as if by understanding mycorrhizae, we could somehow change something about the dynamics of how forests work in order to solve global warming or to reforest the world. And although those are very noble goals, I think it really changed how people went about their research, and it sort of incentivized the certain ways of turning your research into a certain narrative that made people feel hopeful about the utility of fungi in trying to solve some of our big challenges. And that same sort of hopefulness, or same sort of very sort of sweet, fuzzy feeling about mutualistic relationships really was present in Suzanne Simard's memoir, Finding the Mother Tree. And in almost all of her TED Talks, her interviews, her profiles in magazines, you get the sense that fungi and trees and their seedlings are all involved in mutualism all the time, that competition isn't real. And I just want to highlight verbatim one of these quotes from one of her TED Talk interviews, or her TED Talks, is she said, in ecosystems, there is only reciprocity. There is only mutual respect. And in my opinion, this really shows how much a desired narrative can influence your thinking, such that you lose track of the fact that no, it's not actually true that in ecosystems there is only reciprocity and that there is only mutual respect. I think that no matter what you understand about common mycorrhizal networks and about resource sharing, even if there is a lot of mutualism, it is absolutely true that there is also still a lot of competition and a lot of parasitism. And it is not straightforward to assume that just because you see resources moving between two plants, that there was any sort of intentionality about it, or even that it is behaving exactly the way that you think it is. There is a lot more very careful study required for us to determine whether or not these dynamics that we so far have 3 or 4 examples of actually operate the way that they're being talked about in all of Earth's forests. You know, there's a lot of subtleties, a lot of context dependence that we are sort of papering over or ignoring when we take these sort of few narratives we've heard about and we make the assumption that this is how forests work more generally. Some of these narratives show up in places like Ted Lasso. Here's Coach Beard reading Suzanne Simard's books. And if you've read Richard Powers' The Overstory, this was another important place that a lot of people received this narrative about common mycorrhizal networks and about reciprocity between trees and how mutualism works at scale. Suzanne Simard has defended her use of these sort of metaphors by saying she uses this anthropomorphism and these culturally weighted words like mother because it's a way of trying to get people to relate to these concepts, and by doing so, getting them to care more about it. So that was her stated reason for why she has sort of doubled down on these particular metaphors. But I want to show you that Toby Kears, who's sort of a more modern generation of researcher, has done almost the exact opposite of using metaphors of competition and sort of cutthroat resource exchange and very anthropomorphized human economic models to apply to the exact same systems that Suzanne Simard framed as being mutualistic and reciprocal. Toby Kears, in her TED Talk, calls them tiny capitalists, and in fact goes on to say, I want you to think of a market economy that's 400 million years old, so huge that it can connect millions of traders simultaneously, and so persistent that it has survived mass extinctions. Unlike human economies that rely on cognition to make decisions, traders in this market, in this case being fungi, beg, borrow, steal, and cheat, all in the absence of thought. In my opinion, this is going all the way to the other end of the spectrum and imposing way too much human anthropomorphism. But I just want to emphasize that it really is the opposite of what Suzanne Simard metaphorized these systems as, and it relates to the exact same system of mycorrhizal networks. So it shows just how much these things are like Rorschach tests. You can put up the same drawing or the same data in front of different people, and they will impose totally different metaphors about how they understand what's going on below ground. And I think there's a real danger in that. The Society for the Protection of Underground Networks is the sort of project that Toby Kears is most associated with nowadays. And I just want to highlight that in their mission statement up there at the top, it says to map, to protect, and to harness the mycorrhizal networks that regulate the Earth's climate. And it's that word harness that I want to particularly focus your attention on, because this relates to Paul Stamets's 2008 TED Talk, where there's some idea that by understanding or documenting or mapping these systems better, we can harness them, we can somehow use them, we can make them somehow microbial beasts of burden to solve our climate problems. And once again, while that is a noble goal for obvious reasons, I do think there's a danger in assuming that somehow we are going to be able to bend or shape fungi to solve our problems, when really our problems with regards to climate change come from us, from humans, from what we are doing. I think the best we can do there is obviously to protect the existing mycorrhizal networks of the planet, but also probably to limit our consumption, to change how we behave, not to assume that somehow harnessing other organisms will help solve our problems for us. And just to show you how far it goes when you start turning fungal market economies into metaphors, Toby Kears appeared on this podcast about Bitcoin. So people really run away with these metaphors very quickly. And I think it's a real danger to assume that we can turn our, at this point, very slim understanding of mycorrhizal networks into any sort of lesson that we can draw upon for what we do in human markets. I really don't think that's a good place for us to go. And it makes me nervous when I see people who are mycorrhizal researchers ended up in these sort of very peripheral human endeavors like Bitcoin. Paul Stamets apparently recently got into Bitcoin as well. A 2022 European Parliament report on sustainable forestry advocated for the adoption of Wood Wide Web theory into how they manage their forests. And I think it was this kind of thing that really made Justine Karst nervous, because if you haven't gotten the picture from the talk so far, there is no such thing as recommendations of Wood Wide Web theory. There is not one obvious lesson that Wood Wide Web theory teaches us. Wood Wide Web theory is a complicated knot of contextually dependent, subtle, uncertain, barely understood findings that we have not even replicated very strongly across the world. There are many theories, there are many examples, but they're all pretty limited in their ability to be robustly generalized everywhere. So when governments start saying, yes, we're going to take this theory and implement it, it makes a lot of researchers nervous who operate in this world, because they know that there is not an actionable plan suggested by this body of research. And so it was that kind of thing that led Justine Karst to publish her pushback article. And really what she highlighted is that metaphors, the ways that we put human words or human anthropomorphism, human language around this research, can get so strong and so seductive and so powerful as a narrative that it can actually limit where the research goes, because researchers are trying to get funding, or they're trying to communicate their findings to a general public that wants to hear a certain narrative. And it can actually make it harder for us to understand the truth about what the research actually says if we get too committed to a single model of what's going on out there. So I think, actually, that Justine Karst may have pushed back too much for my personal taste, my beliefs, or my understanding about what these mycorrhizal networks are doing. But the important thing that she contributed is that we just need to slow down. and we need to be a little bit more careful as an academic community and as a general public, as science communicators, to really make sure that we are being slow and humble and acknowledging uncertainty where we find it. So this was the New York Times article that summarized Justine Karst's pushback. So thankfully, this is starting to get more widespread mainstream attention that maybe we didn't. Maybe we need to put our feet back on the ground about interpreting the Wood Wide Web. And I just want to show you why it makes science communication so difficult. Here's a headline that I saw in the Washington Post last year. And it says, how a bat disease may have led to the death of more than 1,000 kids. And in the aftermath of COVID, which was thought to be a bat-originated disease, a bat-originated virus, this headline reads like, oh no, there's a new bat disease that may be causing lots of human death. But what it turns out, if you read this article, is that a disease that affected bats killed off a bat population, and the subsequent increase in the insect population, because the bats weren't around to eat them, were transmitting diseases to kids. So it was actually the insects that were transmitting, the mosquitoes that were transmitting this disease, not the bats. And so a headline can go a long way towards influencing public perception of a group of organisms, and whether or not they manage them or relate to them in a certain way can be really skewed from a misinterpreted or misleading headline like this. I think this headline is terrible. Probably worth some sort of, I don't want to say that the journalist should have been fired, but they need a stern talking to about this headline. So science communication, especially in the age of social media, especially in the age that's really hungry for punchy narratives that have catchy takeaway messages, that can really make people, especially researchers, it can make them turn their research into a more tidy story than it actually deserves. And that's actually a really dangerous thing. When science is supposed to be sort of the body of people who are working on understanding the world and all of its complexity and all of its nuance, there may not be a catchy story. There may not be a tidy explanation of how the world works. And so the takeaway that I want to give you is basically that you should not take takeaways. It should not be the case that research comes with a little tidy bow on top. It is, I think, really important to orient ourselves towards appreciating the fact that the world is complicated, and that especially when we're dealing with organisms that live underground, that are microscopic, that operate on timescales that are very short and also very long. Fungi can have effects on their plant partner that happen on the course of months, but a mycelium can also live for thousands of years. That is hard for human beings to wrap their heads around. We are not passing off research projects from one professor to the other for thousands of years. Most of our universities are only 100 years old or less. And to truly understand mycorrhizal dynamics, we're going to have to keep paying attention to these forests for thousands of years to watch the outcomes of certain kinds of manipulation, or certain kinds of, the age of the forest increases, and the dynamics, the relationships of the fungi they're partnered with change over that time. So something that you should also remember is that when we go and we take a sample of forest soil and we tease it apart and we take it back to the lab to determine what's in that soil, you've just destroyed that little bit of soil in the forest. And so you can't watch the outcome of that little patch of forest after sampling it to determine what's there, because you've just interfered with it. So at least with regard to mycorrhizal networks, sampling is destructive, and that makes repeated analysis over time problematic. You have to do some lab experiments, and you have to do some field experiments, and you have to do some observational studies, and you have to try and triangulate between all these different sources of information to try and get a little glimpse, a little bit of understanding about how they all work together. And since a lot of us are millennials, the easiest way to summarize this is through a meme. So this shirt does a pretty good job of it. I want to kiss every bug, but they are so small and my lips are so strong. We are as human beings, kind of big. And fungi are microscopic. At least their fungal threads of the mycelium are buried in soil. And when you go and you try and manipulate them and investigate them and scrutinize their behavior and what they're doing, you actually interfere with that system and you make it so that nothing about that system is now observable in an undisturbed fashion. So it's really difficult to do this kind of research. And that should be something that we keep in mind when we read about mycorrhizal literature, is that it is a very difficult topic. It is not like birds where you can sort of watch them from afar with binoculars and observe their behavior. Fungi are not visible to us in the same way. They're both microscopic and macroscopic, as I mentioned. They're dynamic to the point of paradox, meaning they can be parasites at one point in the life cycle of their host plant and mutualistic with that same plant just a few years or a few months later. And once again, they operate on timescales that are so long that we're going to have to continue doing this research for far longer than any one human lifespan. Research is going to have to be handed down as sort of a research program from multiple generations of investigators before we understand how ectomycorrhizal or mycorrhizal relationships work. Remember that it was only 1881 when we first learned of the existence of this relationship to begin with. We are still in very early days as a human civilization of getting to know this particular kind of ecological dynamic. And so I just want to close with one of my heroes in regard to understanding the philosophy of science. This is Donna Haraway. She was the professor of consciousness at UC Santa Cruz for a long time. She's retired now. And a lot of her writing was just to remind us that metaphor in science is always present. There's no way of taking a purely agnostic perspective from one who has access to the truth without bias. And she reminded us with a lot of her writing that science is done by people, and people have to speak in human language, and that's necessarily limiting and constrained. It comes with its own bias that's always loaded into it. And the stories that we have to tell, we basically have to understand data through story. It is always going to bring in with it some amount of perspective, some amount of bias. And the goal is not to find a perfectly clean, unbiased place to understand science. It's to acknowledge that you're coming in with the bias and to try and account for it and to try and acknowledge that there are other ways of understanding the same kinds of data. And by triangulating between different forms of bias, you eventually get a clearer picture. And just holding your personal metaphors very lightly so that you don't get boxed in by the particular story that you've told. So I just want to highlight this paragraph from Catherine Flynn, who was commenting in the New York Times article about Justine Karst's pushback, her slow down the train article in 2023. Catherine Flynn said, "Between treating fungi as objects or humans, I suggest a third way. Let's seek to understand fungi on their own terms, fundamentally unlike us, mute, rooted, and inscrutable. We need to meet the challenge of cultivating respect for organisms that are different from us, in their separate and complex bodies, in their sophisticated interactions, and in their unfathomable lives." And what I really like about this is that she encourages us to remain in uncertainty, to be okay with the fact that the narratives may not have a tidy ending. They may never end. We may just continue learning complexities, paradoxes, nooks and crannies, context-dependent conclusions that don't hold in every system all the time. And I think that's really important. So I really encourage us to always stay curious about these fungi that we love and the plants that we love. They are remarkable organisms with remarkable interactions that should inspire us. The fact that they can provide a conduit for communication between plants, whether it's in the form of signals or it's in the form of cues, intentional or unintentional. It is remarkable that this happens at all. And for anyone who's an ecological, a student going into a career in ecology, this provides, hundreds of years, if not thousands of years of potential investigations to go and do. You should be very excited as an undergraduate or a grad student going and investigating this world. It's just endless research questions. And as a last quote from Gary Snyder, stay together, learn the fungi, and go light. These are, I think, the most important rules for any naturalist to keep in mind these days. And with that, that's the end of my talk. So thanks for having me here tonight.
Thanks so much, Christian. It's so nice to hear a pragmatic voice in all the noise. And when this all sort of went down around 2023, it felt like it felt like the headline should have been like, "We need more funding for mycology research." Like
Yeah.
that should have been the headlines, but of course that doesn't always, asking for funding doesn't always get people to click.
Right, right. And that's one of the big dangers these days, that sometimes it feels like people are structuring their efforts more for the attention than for the content.
Mm-hmm. Yeah, so we
There's
had
a
some
question
good
here from
questions.
George. Do you know
Yeah.
whether there are pathogenic mycorrhizae? And Tracy responded, seems like an oxymoron. And I totally agree. It feels like an oxymoron, but I think what we've learned is that fungi are sometimes oxymoronic. They can really be paradoxical, where the same fungus can behave like a pathogen at the start of a tree's life cycle, but by the end of its life cycle, it can end up being a mutualist. And it's just that kind of paradox that we really have to remember.
Yeah, so there was another question earlier in the talk about how scientists were able to determine the age of, let's see, yeah, the paleontologists, how they were able to determine, like what methods were they able to use to determine that mycorrhizae was 130 million years old.
That's a little bit above my pay grade, but I think basically the fossil, based on its position in the geologic record, you can use other methods to estimate the age of that rock. And then if the fossil is embedded in that rock, you sort of extrapolate from that the fossil which preserved the structure of mycorrhizal root tips was then assumed to be 130 million years in its origin. But there's probably other evidence out there now that I'm not even familiar with, because I don't do a lot of paleomycology.
Mm-hmm. And let's see, there was a good question. Christina, do you want to unmute and ask your question?
Sure. I know it was kind
I'm
of
reading
long.
it now, yeah.
Yeah. So I read Suzanne Samard's book, and then I'm also just about finished with The Light Eaters by Zoe Schlenker, I think it's how it's pronounced. And she doesn't even touch on mycorrhizal networks at all, which I thought was kind of strange to not address in the book. But she seems to really push that pheromones, that's the scientific research that says that pheromones are how plants communicate dangers to each other, whereas Suzanne Samard said no, it's or I mean not no, but she started with I and maybe I remembered it wrong, but how plants can communicate dangers, like if there's a certain beetle or something eating on them, they can transmit that signals to other plants to say, you know, beef up your defenses or something like that. And I was
Yeah.
wondering if you, in your experience, you think it's a mix of both? Do you think it's one more than the other? I was just
Uh,
curious.
so I am not a plant physiologist and I don't know that much about plant pheromones, but I think I know enough about how ecology works to say that it's always both. Um, Now, not necessarily both for every plant. There may be some plants that totally lack the kind of pheromone signaling that she's talking about, and there may be some plants that are especially good at that pheromone signaling. And I think the sort of usual general answer is that it depends. It depends on which plant you're talking about, which system you're talking about, what pathogen you're talking about, what stressor you're talking about. But it would not surprise me at all if many plants are using a mix of those strategies for information detection. You know, if you have both, why would you ever not use both? Um, and I could not begin to say if it's more one or more the other in any system. I don't know enough. But yeah, it is sort of odd that in that book, she wouldn't mention more about mycorrhizal networks, because I feel like almost everyone else has sort of put that front and center of their understanding these days.
Mm-hmm.
Thanks for that question, Christina.
Yeah, and you had touched on too, like the kind of the limitations too. That's kind of where I always sort of like get stuck is like the limitations of our technology and where we are in science, like and the ability to like look at things in situ, and then also even um, in the field, like we have such limitations of
Yeah.
what we can look at once. And I know that like in Peter McCoy's book, he talks about, endophytes, like both bacteria and fungi, like that being kind of like the microbiome. And so we get into this, especially within like Western science, we have these like limitations, and it's so based on kind of putting everything in its box and its classifications that we think like, okay, well, let's just look at these mycorrhizal fungi and not like the whole biome, because it's
Right.
all working together, because we can't really within the limitations of our current practices within science. So, I and there's I've seen these debates too around kind of like the way we speak, the sort of the language that we choose to talk about plants too, because there's such a movement too to sort of have more relationship or like integrate more into nature, like through traditional ecological knowledge and like traditional ways of knowing.
And
There's that.
I would like to address that specifically, which is that, Sarah says we need to keep a taut rein on anthropomorphizing plants. And Angel, like you just mentioned, there is this increasing resurgence of traditional ecological knowledge and having sort of relationships with plants that are often highly anthropomorphic. And
Mhm.
I just want to make it very clear that I love anthropomorphizing fungi and I love anthropomorphizing plants, and that's actually kind of my favorite thing to do these days. And what I'm not arguing is that there is no place for
Mhm.
anthropomorphizing other creatures. And in fact, I think it is absolutely required because it's
Mhm.
really hard to understand anything without the use of human language metaphors that make it sort of intelligible
Mhm.
and communicable between people. What I am saying is that we need to be aware of that metaphor, be aware
Mhm.
of that anthropomorphizing, and we need to be very honest about where it runs out, like where we can't use it anymore, where it's
Mhm.
not an appropriate way to plan action. Now, when you're having a personal relationship with a plant or where you're learning about it from a traditional ecological knowledge perspective, it can often be an, a very powerful way
Yeah.
of learning about the world. Um, but it can also be very misleading, in the scientific
Mhm.
world to assume that what counts as evidence can ever
Mhm.
be addressed by the technology that you have. Like if you are limiting yourself to the Western tradition of science, there are some kinds of evidence that will be impossible to get because you have said that doesn't count as evidence. So
Mhm.
it's a very complicated and very subtle thing that requires a lot of self-control, a lot of patience, and a lot of humility. And
Right.
unfortunately, media, narratives, social media, and funding really don't reward humility,
No.
self-control, and patience.
Like it just doesn't really fit into the paradigm where, a researcher goes to university for a couple of years, and kind of how you were saying, like you need to like spend, and that's where like traditional ecological knowledge comes. It's like intergenerational. It's like multi generations of knowledge getting passed on. And we don't have that so much. We have like digital nomads, like people moving all the time,
Right.
like fast paced world, like, people with 3 jobs, like it's impossible to like have that like sit spot, like that
Right.
time and place based knowledge of our organisms. And so it's sort of like the Western science just does not allow for it in a way, like
Yeah.
unless
Western civilization. Yeah.
yeah.
The whole Enlightenment way of constructing the world around rationality really starts to divorce people from uh, the sort of relationships we evolved with, which was sort of like
Hmm.
having a home base that you probably saw your entire life. Imagine knowing
Hmm.
one tree your entire life. Um, and it's really easy to forget that for almost all of human history, people lived around the same set of individual organisms for all of their time that they were
Hmm.
alive. It's
Hmm.
only very recently that we've had I'm going to put this in quotes, luxury of ignoring the organisms around us.
Hmm.
It's not really a luxury, in my opinion, but it is coded under convenience and luxury that we are able to sort of put that much distance between our material ecological surroundings and our daily life.
Hmm.
So no wonder we don't understand things very well.
Yes, yes. Well, all great conversations. Do we have some other questions? And feel free if people want to come unmute.
Yeah, I want to be mindful of our people's time here. I know we're running over time, but
Yeah.
oh,
I had
George
a-
asked. Oh,
I had a question.
is this
I
George?
don't know. I can just butt in.
Yeah, go ahead.
I guess, and it's also just kind of more of an observation. I'm Art. Involved with the Mycological Society and occasional instructor and whatnot. But and I'm self-made, like many of the people in this field, and I feel like a lot of the slippery slope of the anthropomorphizing is it directly kind of in direct relationship with the novel sort of field that we're all kind of, finding ourselves in. And also because it's so complex. There's so many different, kind of what we were talking about in the beginning, is so many different ways that somebody can approach mycology from completely different angles and inspirations and motivations and whatnot. And so I feel like a lot of that is gets sort of jumbled when you see these headlines or you see, I mean, you're on TikTok or on Instagram. The ads on Instagram are mushrooms are going to cure everything. And because we're so kind of quick to sort of consume these little sound bites and whatnot and kind of rely on them, hang our hats on them, that's where I feel like it's almost tantamount that we kind of self-police a little bit, not to the point of making it drone on and on about scientific dialogue or terminology, but just I guess it's I kind of want to get your take on how, like, since let's just say since Paul Stamets after Paul Stamets PS his TED Talk, how you feel like information is sort of shared, verified, in this kind of post-hype world where we're all in it and we're all kind of diving in and people are buying mushroom coffee at the grocery store and whatever it is, we're not even really knowing what it is. So
Yeah,
I'm kind of
I
rambling,
mean,
so I'll let you go.
I think what you said there, the self-policing inside the community, um, and obviously that comes with some pejorative connotations these days, but let's just say uh, I like the idea that being able to hold each other accountable for that we propagate out into the world from the mushroom community is is really the place to start. And I'm going to just share your fealism and overwhelm about the way that information is passed around in the world these days, because it is getting really hard out there for people to understand what's true about anything. I mean, the world is so flooded misinformation, both intentional and unintentional, or failures of design. But so many people I know who I thought were sort of a little bit more sophisticated, a little bit more immune to fake information or like even just AI generated sort of spammy, um, seemingly like sciencey type research summaries. It's getting out there for unsophisticated people or beginners to know what's true. And I think there is a real responsibility that we have as people who are inside the mushroom community a little bit more to be sort of sources of constraint for our neighbors and for the people we talk to. Just be like, you know what? You may have heard this, and when people say things to you, to not just foster their excitement, but to respond to their excitement with also additional curiosity in the form of skepticism about whether or not they know why they know. It's like, okay, you may have heard something, but why do you think that that's true? Not to deny what they're saying may be true, because it may be, But just to really encourage the people that you know to take the extra step of getting down to the bottom of where a message came from, what conclusions it actually leads us to. I think that's going to be an extremely important skill to cultivate as humans generally about everything we do for the foreseeable future.
Yeah, I agree, especially with that your last sentiment. And I'll be honest, I didn't know much about your talk. I just saw the clickbait headline, and came on. So I wasn't really sure which way you were going to go.
That's
And
good. I like to keep you guessing.
yeah, and I'm pleased that you sort of showed us both ends of it, because I think a lot of people who are keepers of the faith or skeptics or whatever will look at Toby Kears and just believe whatever she says because she's put in the work.
Yeah.
But at the same time, so has Paul Stamets, and he's like you said, he's said some pretty irresponsible things in front of millions of people. So yeah. Anyway, just thanks for all the insights. It's been
Yeah, sure
fun.
thing.
Yeah.
Thanks for tuning in.
Yeah. Anyone else? I did see George asked a question. Do you know whether there are research done on mycorrhizae and urban trees, particularly those planted in crappy or engineered soil?
Yeah, so there is a lot of research. Well, more now, a lot, but there is an increasing amount of research on mycorrhizal networks in urban environments. Although I will say, in many cases, it's hard for the network to even develop because oftentimes things like concrete or urban infrastructure isolate trees from each other, so that the formation of multi-tree networks, common mycorrhizal networks, is prevented just by the structural features of the urban environment. But there are people studying which mycorrhizal fungi are able to tolerate those crappy soil urban environments better than others, and which provide a greater advantage to their host in those kind of settings. But it sounds like that's, I assume some of what Ellen's doing, some of the people she's working with, would know more about that and would be interested in figuring that out. But there are certainly mycorrhizal fungi that are able to grow with urban trees, depending on the identity of the tree. Not every plant can do this. So if you have nice urban oaks or pecans or things like that, then yeah, you can definitely get some mycorrhizal fungi in those settings. But not every fungus is able to make a living in those urban environments.
Great. Well, I think that wraps up all the questions. Thank you for staying on a little bit longer, and
Sure thing.
we appreciate it. Appreciate you sharing your very middle of the road, the green zone.
That's me.
That part of the rainbow.
I'm an extreme moderate.
So yeah, everyone have a great night, and thanks for joining us, and definitely come out and help us build some Johnson Sioux bioreactors in April.
That sounds like such a cool event. I wish I
Yeah.
All right. Bye, everyone. Thanks for
Bye,
having me,
everyone.
Angel. Thanks to the
Thank
Central
you.
Texas Myco Society.